Hello, this is Yashveen. In this video, I'll be solving paper 2 May-June 2019 and 700 biology. 1 Figure 1.1 is a diagram showing the structure of a section of a DNA molecule. A, draw a circle around one monomer of DNA in 1.1. So a monomer of DNA would be a nucleotide since DNA is the polymer. Now be circling this bottom left nucleotide over here. You can circle any one. B, name the two bases forming the base pair at X and give a reason for your answer. So these this base pair of the two nucleotides is of cytosine and guanine, since there are 3 hydrogen bonds represented by the 3 dotted lines between the two bases. C, the statements 1 to 5 describe the events that occur during DNA replication. Write the numbers 1 to 5 in the spaces below to show the order in which these events occur, and the first one has been done for you. So the first step is hydrogen bonds breaking between the two strands of DNA, and then the two strands of the DNA double helix separate so that each one of them can be used as a template. And so the next step after 3 would be 5. After that, DNA polymerase forms phosphodiester bond to form the new strands, and then hydrogen bonds form between complementary bases. And finally, DNA double helix form. D, the telomere is a region found at the end of a chromosome. Outline the function of telomeres. So these would be repeated lengths of DNA that do not have any function other than to preserve the entire genetic information. So they prevent loss of genes and allow continuous successful replication. Figure 2.1 is a photomicrograph showing a transverse section through a bronchus and the surrounding lung tissue. A, calculate the actual diameter of the lumen of the bronchus using the line PQ, this line over here, and you're supposed to write down the formula you used to make your calculation, show your working, and give your answer to the nearest micrometers. So the formula obviously would be magnification is equal to image size over actual size, because of which actual size is image size over magnification, or image size divided by magnification. Now the image size in this image is around 70 millimeters or 7 centimeters. Convert that into micrometers, which would be 70,000 micrometers, and then you divide that by 40 and your answer would be 1750 micrometers. B1, in figure 2.1, the area labeled R shows a section through some alveoli. Name the tissue that lines the air spaces of alveoli. That would be squamous epithelial, which makes the alveolar walls. To suggest how the alveoli are adapted for gas exchange, they have a number of features for that. They have elastic fibers in their walls that can stretch and recoil for inhalation and exhalation. This also increases the surface area for diffusion of carbon dioxide and oxygen across the alveoli and capillaries. And their walls are only single cell thick, which allows quick diffusion of the gases. See, in the leaves of plants, both gas exchange and transpiration occur through stomata. Suggest how the arrangement of cells in the leaf of a dicotyledonous plant contributes to the loss of water by transpiration. So gas exchange would be exchange of carbon dioxide and oxygen, whereas transpiration would be evaporation of water vapor from cell walls of mesophyll cells into the air spaces, followed by the diffusion of water vapor down the potential gradient through the stomata. And you're supposed to describe how the cell's arrangement favored the transpiration process, the entire process of evaporation and then followed by diffusion of water vapor. That is aided by the fact that large spongy mesophyll cell walls have a large surface area which allows water vapor evaporation into the air spaces, and also that stomata are arranged below the air spaces, which then allows diffusion of water vapor after they have been evaporated into the air spaces. So the stomata are just below the air spaces. The figure 2.2 shows the mean transpiration rate of a xerophyte between 8 am and 7 pm. So you've got this graph. We've got mean transpiration rate on the y-axis and the time of the day on x-axis. One, describe the data shown in figure 2.2. First off, the rate of transpiration increases to the maximum at around 9 am to 5 units, and then the rate decreases at 10 am, and then from 10 pm to this time over here, which is 3 pm, the rate of transpiration fluctuates, after which it falls down to 7 pm at the lowest rate, which is 0.5 units. Because the gradient of the graph did not change while the rate increases, I've only mentioned that it increases. However, while the rate decreases, the gradient of the graph changes. The rate is steepest between 3 pm to 4 pm, and then the rate becomes slower. I've written in my answer that the steepest decrease in transpiration rate occurs between 3 pm and 4 pm. To suggest why the transpiration rate changed between 9 and 10 am. So between 9 and 10 am, the rate of transpiration decreases, and so you can write about the factors that can cause decrease in transpiration, which could be that the humidity around stomata has increased and maybe the wind speed has decreased. Or you could write about the light intensity and temperature having decreased. All of these would reduce the potential gradient, which in turn reduces the rate of transpiration. Cholera is an infectious disease caused by the bacterium Vibrio cholera. The pathogen causes disease by disrupting the activity of the epithelial cells in the human intestine. A complete table 3.1 to compare the structure of V. cholerae and an epithelial cell from the human intestine. Use a tick to indicate the presence of the structure and a cross to indicate the absence. The first row has been completed for you. So the cell wall is present in the pathogen and not in human epithelial cell, because V. cholerae has a structure similar to that of a bacterium, and so it would have all the features of that. Both of them would have a cell surface membrane, ribosomes as well, and a large permanent vacuole is only present in a plant cell, and so it would be absent from both of them. And organelles surrounded by a double membrane are present in an animal cell and not in that of the pathogen. The pathogen would have 70S ribosomes and the epithelial cell would have 80S ribosomes. B. Describe the methods that can be used to prevent the spread of cholera. Alright, in this answer you need to write down initiatives that can be used to break the transmission cycle, which could mean using vaccines for the disease so that a person receives active immunity and does not get infected, and setting up sewage disposal sites or water purification tanks, which would ensure that the water to be used is not contaminated and infected sewage does not come into contact with food or water to be ingested. You could also write about providing oral rehydration therapy as a treatment and educating people about washing hands before eating and not handling cooking utensils without doing so. See, the WHO has suggested that people with HIV/AIDS take a longer time to recover from cholera and are at an increased risk of death from cholera. That is because the HIV/AIDS attacks the immune system, which is supposed to help the body fight all sorts of diseases. The human immunodeficiency virus only affects certain types of cells. These cells have CD4 receptor proteins in their cell surface membrane, helper T lymphocytes have CD4 receptor proteins, or in other words, the HIV only affects cells like T lymphocytes, as it has receptor proteins that are complementary to the glycoproteins present on HIV. One figure 3.1 is a diagram of HIV showing the glycoprotein GP120. The glycoprotein is embedded in a membrane envelope which surrounds the viral protein coat. So you've got the genetic material protein coat and an envelope which has got GP120, and this is probably going to bind to the receptor proteins present on the target cells, which are T lymphocytes, and the receptor proteins would be CD4. The glycoprotein GP120 is important in allowing HIV to only infect certain types of cells. These certain types of cells are supposed to have the receptor protein CD4, and you're supposed to suggest the role of GP120. So this GP120 is going to be complementary to CD4, and it's going to bind with it on helper T lymphocytes to infect them, because for the binding to occur, the shapes have to be complementary. The antigen and the receptor are supposed to be complementary. Do people with HIV or AIDS have a lower T lymphocyte count? Explain how a lower T lymphocyte count could reduce the body's ability to produce antibodies against V. cholera. So there are two types of T lymphocytes, helper and killer T lymphocytes. As the name suggests, killer T lymphocytes kill infected cells, whereas helper T lymphocytes secrete cytokines, which one stimulates phagocytosis and two stimulates the B lymphocytes to form plasma cells and secrete antibodies faster. And if the T lymphocytes are targeted in this disease, that means the only antibodies produced would be using the plasma cells or the B lymphocytes that have recognized the HIV as a foreign antigen. And so no B lymphocytes would be stimulated by T lymphocytes, helper T lymphocytes, and therefore a fewer amount of plasma cells would form, resulting in a fewer amount of antibodies produced. Four phloem sieve tube elements and xylem vessel elements are cells that are involved in the transport of substances in plants. A. Describe two differences between the structure of a plant phloem sieve tube element and a xylem vessel. So phloem sieve tube is adapted to transport assimilates, whereas xylem has structures that support the transport of mineral ions dissolved in water. So phloem sieve tube has some amount of cytoplasm, whereas xylem has none at all. And phloem sieve tube also contains sieve plates with sieve pores between them, whereas xylem is made up of dead cells whose end walls have disappeared, so there are no sieve plates in xylem. B. Sucrose is transported in phloem sieve tubes. A student carried out a test to identify the presence of sucrose in a sample of sap taken from inside a phloem sieve tube. One of the steps in the procedure instructed the student to heat the phloem sap with hydrochloric acid. Explain what is necessary to carry out this step in the procedure. Hydrochloric acid can break down sucrose to reducing sugars, and then their presence can be tested by a Benedict's solution, whereas we don't have a test for sucrose on its own. See the process of loading sucrose into a phloem sieve tube element. Involves a companion cell. Figure 4.1 shows a stage in the process of loading sucrose into the phloem. So you've got the cell surface membrane of companion cell and its cellulose cell wall. In this step, the hydrogen ions are transported from the cytoplasm into the cellulose cell wall against their concentration gradient by active transport. So it requires ATP molecules to provide the energy. Companion cells contain large numbers of mitochondria and ribosomes. Explain the roles of mitochondria and ribosomes for the stage shown in figure 4.1. So mitochondria obviously is required to provide energy or ATP for active transport of hydrogen ions, and ribosomes would be required to produce or to synthesize the transport protein which is required to carry hydrogen ions from the cytoplasm into the cell walls. Since the transport is active transport, and so it requires a transport protein. To describe and explain how sucrose is transported from the cell wall of companion cell into the cytoplasm. Now that a hydrogen ion concentration gradient is created between the cell wall and the cytoplasm after their movement into the cell wall, it is transported by facilitated diffusion back into the cytoplasm of the companion cell along with sucrose molecules using co-transporter proteins. 5a figure 5.1 shows a photomicrograph of human blood cells that have been placed in a solution of sodium chloride. 1. State the function of the cell labeled Y. So Y is a neutrophil since it's got lobed nucleus. As it is a phagocyte, it is supposed to carry out phagocytosis. 2. The red blood cell labeled in figure 5.1 is described as crenated because it has an abnormal shriveled appearance. Explain how this red blood cell has become crenated. So it's become crenated because the water potential of the solution that it has been placed in is lower than the water potential inside the red blood cell due to the sodium chloride compound that is present in the solution, and as a result water moves out of the cell by osmosis, giving it shriveled appearance. B. Researchers studied the effect of altitude on the red blood cell count of humans. They recorded the red blood cell count of healthy adults from populations living at 4 different altitudes in a mountainous region, and the results are shown in table 5.1. 1. Calculate the percentage increase in mean red blood cell count between population A and population D. So in this column you've got the mean red blood cell count. Between A and D, you're supposed to calculate the difference. So you subtract 4.5 from 5.1, divide that by 4.5 and multiply the answer with 100, which would give you the answer as 13.6, correct to 3 significant figures. 2. Describe and suggest explanations for the data in table 5.1. So going from populations A to D, the altitude is continuously increasing, where the red blood cell count only fluctuates, increases and decreases. And in the table, population B at altitude 1676 meters has the greatest mean red blood cell count, and A has the least red blood cell count at 695 meters. This could be because of the fact that at higher altitudes the partial pressure of oxygen is lower and less oxygen is inhaled in the lungs. It decreases the percentage saturation of hemoglobin with oxygen as its affinity for oxygen has decreased. C. Carbonic anhydrase is an enzyme found in red blood cells which has an important role in the transport of respiratory gases. Explain why a non-competitive inhibitor of carbonic anhydrase will reduce the supply of oxygen to actively respiring tissues. Non-competitive inhibitor would bind to allosteric site of enzyme and change its tertiary structure, and this prevents the formation of hydrogen carbonate and therefore the formation of hydrogen ions and carbonate ions. Now, hydrogen ions are required to displace oxygen molecules from oxyhemoglobin so that oxygen is supplied to respiring tissues where the partial pressure of oxygen is lower, and if the inhibitor binds to the enzyme, hydrogen carbonate and therefore hydrogen ions are not formed. Therefore, there is no substance that would be able to displace oxygen from oxyhemoglobin, and therefore oxygen would not be supplied to respiring tissues. 6. Figure 6.1 is a diagram of a vertical section of the human heart and the associated blood vessels. A completes table 6.2 by writing the letter from figure 6.1 that identifies each of the structures described. So the valve preventing backflow of blood into a ventricle. But blood could flow from arteries into ventricles, and that is prevented by semilunar valves represented by this valve over here S. This backflow of blood into ventricles could have occurred when the pressure in ventricles falls below pressure in arteries, so that would be after ventricle systole. The second row asks for the blood vessel carrying blood under the highest pressure, so that would be aorta, which is P, and the chamber that pumps blood into the lungs, which would be right ventricle, and that is N over here. B. Describe the short-term effects of nicotine on the cardiovascular system. Nicotine has a few effects, including it increases heart rate and blood pressure and risk of blood clotting. C. Cardiac myocytes are heart cells. Fetal cardiac myocytes are present before birth and divide rapidly during fetal development. Soon after birth, the cell cycle in most of these fetal cardiac myocytes stops, forming adult cardiac myocytes. Most of the adult cells have completed the cell cycle, but in some cell cycle stops at the end of mitosis. So the cell cycle is not completed, since the complete cell cycle stops at cytokinesis, which occurs after mitosis. And so in some of the cells, the cell cycle has not been completed. One, state the difference between adult cardiac myocytes that have completed the cell cycle and those that have stopped at the end of mitosis. So if the cell cycle has been completed, the two daughter cells would be completely normal. Whereas if the cell cycle has not been completed and it stops right after mitosis, that means the cell has two nuclei, since the cell has not divided to form two daughter cells. To suggest why it could be a disadvantage that the cell cycle has stopped in most cardiac myocytes. If the cell cycle has stopped, that means no more cells can form that are specialized to function as cardiac cells, which also means that these damaged cells cannot be replaced, and therefore cardiac tissue cannot be repaired. Replacing damaged cells and repairing damaged tissue is a function of mitosis, and therefore this question also answers for drawbacks of cells not being able to carry out mitosis. Alright, this is it. We are done with this paper. Thank you for watching.